This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 477, recorded on January 19th, 2018. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And it's kind of a mild winter day here. It's 33 Fahrenheit, just above freezing, and light wind, high overcast, and not unpleasant.
It's 3 Celsius here. It's kind of interesting high clouds. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
Very well.
We got cloudy 48 degrees Fahrenheit, 9 degrees centigrade. It's what it is. I'd rather sunshine and 60. But actually, we had another 3 days of three consecutive nights of freezing temperatures. So we're coming out of that, sending it your way.
Thank you.
Enjoy it.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. Here, it's sunny. It's beautiful. It's 33 degrees Fahrenheit, 1 degree Celsius.
Kathy has just pasted into the show notes a lovely photo of a plate of cookies.
Yes.
So many different kinds. Did you make these?
I made some of them, and some of them were from the Cookie Exchange.
Some pretty nice cookies here.
Yeah.
Now, you didn't make the swirly ones, did you?
I did not. Christiana made those.
Ooh.
And if you notice, there's 2 kinds, white outer swirl and chocolate outer swirl.
Yeah, that's right.
Oh, yes.
This year's cookies had a very chocolatey influence to them.
I like the Linzer torts. Those are great. Did you make those?
Yes, I did make those.
I love Linzer torts.
Yeah.
Yeah, beautiful. Making me hungry. Dickson will be with us shortly. Kathy, any news about ASV?
Yes. Did I recalculate this from today? Anyway, abstracts are due February 1st. And that's when travel award applications are due. And Andrea in the ASV office is getting swamped with people just now updating their memberships. So if you send her a question, you'll have to understand that you're in a line of a bunch of other people that didn't do it until now. But I want to just encourage people in a positive note. It's a really terrific meeting program. There are scientific sessions. And of course, ASV is known because we have a lot of good career sessions. There are options for lunches for trainees to have with speakers and other scientists at the meeting. Of course, there's a live TWiV. You can find things by going to asv.org or to the meeting site asv2018.umd.edu. And the meeting is July 14th to 18th. 2018, get your abstract ready and submit it.
You got it. 11 days. You had recalculated.
Okay.
All right. We have some follow-ups. David writes, Dickson cited a poem about a candle burning, and this triggered the following memory. And this is a poem. My candle burns at both ends. It will not last the night. But ah, my foes, and oh, my friends, it gives a lovely light. By Edna St. Vincent Millay in 1918, which of course reminds me of Spanish influenza. Yours sincerely, David. P.S. Although Dickson's poetic moment in one of the last TWiVs brought this to mind, Edna appears to consistently have been called Vincent by her friends. Good for you. Yep. Yes, but I'm not very poetic, although I like poems. Hey, Rich, take the next one, please.
Mark writes, Dear TWiVodrome, this email has 2 parts. 1, follow-up to TWiV number 476, and 2, a listener pick of the week. So here's the follow-up. This is great. This is a new, unique salutation that is TWiVodrome. As I write this, Google, the expert, shows no prior usage of this term. Trust but verify. Searches on Google with either search string TWiV-odrome cite Microbe TV or TWiV, no dash, Odrome, Site Microbe TV, reports no hits, i.e. no prior usage.
All right.
I am a fan from the Bay Area in California. Our state is being hit hard by a variety of disasters, fires and floods in Southern California, and high incidence of flu cases and deaths all across the state. Sadly, the twin avoidance ideologies of I will never get the flu or flu shots give you the flu, coupled with the mismatch between this year's flu vax vis-a-vis viruses in circulation has hit this state and others really hard. This writer believes in flu shots. 2017 was the 22nd year in a row both my wife and I received them, and I'm pleased to say we have been flu-free. I often spend time in prime venues for exposure to flu virus, airports, hotels, and in sealed aircraft, breathing the recirculated particulates from everyone who coughs or sneezes. Would any of the TWiV hosts disagree with the claim that airplane air should be branded as a fomite? It's not just door handles, other people's hands, or the doc's stethoscope that spreads infection. And he's got a listener pick that was moved below. So this, I think, comes heavily in the area of pedantry. And Wikipedia defines a fomite as any non-living object or substance capable of carrying infectious organisms, hence transferring them from one individual to another. Skin cells, hair, clothing, and bedding are common hospital sources of contamination. Blah, blah, blah. You know, I mean, I've been teaching about fomites for God knows how many years. I've never considered the air as a fomite, you know. That's like, you know, I mean, it is a non-living object. Is it an object? Sure. I mean,
it's a substance, right?
It's a substance, yeah. So, you know, formally, I guess air is a fomite, but I've always thought of airborne transmission as sort of a different category than transmission by fomite.
Yeah, I think when we talk, I think the purpose of fomite is to distinguish a route of transmission.
Yeah.
And if we classify air as a fomite, we'd have to classify water as a fomite. And then everything is just fomite transmitted. And so what's the point of the term?
Yeah, I think the original intention was like blankets and objects, right?
Doorknobs. And,
a different way of transmission. So I would agree with that.
Formally, you can make the argument, but that's not how it's used. How about that?
Yeah, I mean, for teaching purposes, I think it's clearer if we call it not a fomite, but just transmission route. Yeah. Alan, the next one you should read.
Yes. Anthony writes, here are a couple of pictures that I took of the stealth bomber doing a flyover during OpSale 2000. I was in Liberty State Park taking photos of people and ships on the river. I had not brought a telescopic lens, and light was not the best. And sends a picture, really cool picture of a stealth bomber flying right past the Twin Towers.
Amazing. I tell you.
Back when there were Twin Towers and you could do a low flyby over New York without everybody panicking.
Yeah, it looks photoshopped, but I believe Anthony that he took.
Oh, yeah.
Doesn't it look photoshopped?
No. I mean, well, I suppose it looks photoshopped only in that you don't really expect to see a stealth bomber flying over the Twin Towers.
Well, also, that aircraft looks photoshopped no matter where it is.
Yeah, the one over the Rose Bowl last week, which I think inspired this, right? That looked photoshopped as well.
Right.
Because this bomber is...
Because it's dark, flat black, and it's going to be silhouetted against anything that it's against. And so...
Thank you, Anthony.
So we now have a stealth bomber arc. Wonder how many pictures.
But you won't be able to tell when that's going to come up.
That's right. I meant to mention about influenza. Mark was talking about influenza. This is a very popular vaccine to say I never get the flu vaccine, but this year is a very serious year. Maybe next week we'll have a kind of summary of what's going on. On TWiP this week, Daniel Griffin, our resident infectious disease physician, said he's seeing a lot of people with serious flu this year, and he said a lot of them are going to die, and most of them have not been vaccinated. So he said very healthy people who don't feel they should be vaccinated getting hit hard this year for whatever reason. So you should get the flu vaccine. There's really no reason not to. $10? Come on. No.
A lot of places, it's still free.
Yeah.
If you're over 65, it's going to be free. If your town has got a well-funded clinic for it, it's going to be free.
And don't say you're supporting Big Pharma. That's not the reason to get your flu vaccine.
Yeah. I don't think Big Pharma's making big money on this anymore anyway.
Profit margins on this are really, really slim. They do make money on it, but they also spend a lot of money on it.
The profits are thinner than an eggshell.
Yes, there you go.
I was muted because I was typing, so I didn't get to interject in time back on Mark's comment about all these, I never get the flu shots give you flu, and so forth. There was a really nice blog post in The Guardian addressing this, which the title is "Don't Knock the Flu Jab, It's a Modern Miracle." And I came across this on Twitter because Adam Loring tweeted it with some additional stuff that was very complimentary to the article, too.
There's also an article in the Times this week. I think it was something like flu vaccine. It's not just about you.
Yes.
Oh, that's good. Yeah.
Put that one in as well. Okay, now we have a duo of papers that were published back to back, and a number of people sent in emails about these. Fernando wrote, not open access, so I just read the abstract, but it seems to fit into the retrovirus-derived function ARC. Retro machinery enables neuroplasticity. James writes, Vincent and fellow TWiVologists, I'm a committed listener to TWiV and other associated podcasts. I came across this summary of NIH-funded research, which appears a TWiV natural. Virus-like transfer of mRNA among neurons. What could be better? Keep up the excellent science communication you all support. I'm in wonder of your scheduling abilities and commitment. So am I. I'm amazed that everyone says yes. James is a supervising toxicologist in California.
Cool.
And Chaim writes, here's a really cool new paper in Cell describing a gag-like protein from a retrotransposon that still functions in ways reminiscent of a viral capsid, important for synaptic plasticity. Appears to be captured multiple times like the placental envelopes in TWiV 472. Thanks for all you do. So we have two papers, and it turns out they are different. So they're different enough, so we should do both of them. We'll do two snippets. How's that?
But good luck making a snippet out of either of these.
I was just going to say, good luck with that.
Well, I don't want to do every experiment, but we'll give a feel. One is mammalian and the other is Drosophilin. One is retrovirus-like gag protein. ARC1 binds RNA and traffics across synaptic bouton. This comes out of the University of Massachusetts at Worcester. The first two authors who contributed equally are James Ashley and Benjamin Cordy. Then we have Deandra Lucia, Lee Fradkin, Vivian Budnick, and Travis Thompson. And the other paper, these are both in Cell back-to-back, is the neuronal gene ARC encodes a repurposed retrotransposon gag protein that mediates intercellular RNA transfer. Intercellular, I just thought of Interstellar, right? That's the movie. And the authors here are Elissa Pastuzin, Cameron Day, Rachel Kearns, Madeline Kirk-Smith, Andrew Taibbi, John McCormick, Nathan Yoder, David Belknap, who I have co-published with, Simon Erlandson, Dustin Morado, John Briggs, Cedric Fischot, and Jason Shepard from University of Utah and the MRC Laboratory of Molecular Biology. The ARC.
And University of Copenhagen.
University of Copenhagen. Oh, there it is, stuck in there. Eluding. Yes, Kathy, what were you going to say?
I was going to say that, interestingly, the in brief that's on the cover page of each of these is identical. So I suspect that Cell writes those and gets to plagiarize itself. So in brief, the neuronal protein ARC is evolutionarily related to retrotransposon gag proteins and forms virus-like capsid structures that can transfer mRNA between cells in the nervous system.
All right. So
great. Let's do some email.
Okay, that's
All
it.
right, we're done.
Yeah, that's basically it. So there are two parts of this.
And the authors on these papers were obviously made aware of each other, and each paper comments on the other papers fit.
So there's two parts to this story. One is ARC, which one paper defines right away as activity-regulated cytoskeleton-associated protein, ARC, A-R-C. And the other paper, as far as I can tell, never defines what ARC stands for. Nevertheless, ARC is, and by the way, Kathy, I think you'll agree with this, that one paper calls the neuronal gene ARC, which of course it is not.
Right.
Because the ARC is in every cell, as far as I know. Maybe expressed only in neurons, right?
Express gene, yes.
It is really important for many aspects of CNS function, a master regulator of synaptic plasticity in mammals.
Which I had to look up.
Tell us what it is.
Oh,
you don't want to tell it?
Oh, it's here in a Wikipedia link. I've got it. The ability of synapses to strengthen or weaken over time in response to increases or decreases in their activity. Since memories are postulated to be represented by vastly interconnected networks of synapses, synaptic plasticity is one of the important neurochemical foundations of learning and memory. So I guess that's why this is a big deal. This is potentially going to affect learning and memory.
So the way I look at it, my synapses are not plastic anymore. Welcome, Dickson.
And mine are made out of cement.
You know about synaptic plasticity?
I do.
All right, we just started these two papers.
Right.
And so ARC gene is
gag-like protein.
Is really important for it. And it is also important. So long-term memory, as Kathy said, it's also been implicated in neurological disorders, including Alzheimer's disease, neurodevelopmental disorders like Angelman and Fragile X,
right,
and schizophrenia.
Oh my goodness!
So this ARC is important. So these things are known about ARC, but not much is known about its biochemistry and its function. And that's what these two papers address. One paper, I think, let's do first the University of Utah paper. This is the mammalian, and the other addresses the Drosophila homologue. The other part of this.
But before we get into the papers, could we just for the Caleb's out there describe why we think why this is virology, what a retrotransposon is, and what gag is.
It's kind of it's a little distant from virology. So the other part of the story is retrotransposons, right? And LTR retrotransposons. These are a kind of transposable element, DNAs that can move across to different parts of the same genome. It can be in
jumping genes,
right? Now, LTR retrotransposons, two LTRs, long terminal repeats, which are also characteristic of the retroviral DNA copy. Right. And then they typically encode what we call a gag protein. The gag is an old word from years ago when we didn't know a lot and we called things as we saw them. It means group specific antigen, right? That Rich and Kathy will know that group antigen because they're using antibodies and they found this antigen that was common in cells infected with different retroviruses. And now we know it's the precursor for structural proteins of retroviruses, like the capsid protein. Retroviruses have a capsid that contains the RNA, and then they're wrapped in a membrane. It encodes the protease. It encodes the nucleocapsid that coats the viral RNA and other proteins needed for budding. And did I say reverse transcriptase? So that's the important part of retrotransposons, where the retro part comes, because these are produced as RNAs from the genome, and then the RNAs are copied by reverse transcriptase into DNA. Then they go somewhere else and integrate in the genome. Now, if you take a retroviral gag protein and produce it by itself in a cell, it will make particles, and those particles can be released from the cells, but they're not infectious because they don't have envelope proteins. Now, many people believe. Or not believe. There is evidence that, because science is not a belief system, there's evidence that LTR retrotransposons are actually predate retroviruses.
My goodness.
They're around first.
Retroviruses.
Right? So, and then somehow they acquired an envelope and they became able to travel from cell to cell and they became viruses. Okay. So in a sense, this is not, there is the other possibility that retrotransposons are degenerated retroviruses, but there is less phylogenetic support for that. Okay.
So GAG is like a capsid protein coming from these retrotransposons, ultimately evolutionarily destined for retroviruses. And
but they're also in the cell, right? There are lots of them in the cell.
They're also in the cell. And this gene that we're going to talk about looks like a repurposed GAG protein.
That's right.
Okay, so that's the connection to virology.
Right.
So it exists in the genome just as a gene, this ARC gene. It has sequence elements of GAG, but there's no. It's not part of a LTR retrotransposon. It's by itself.
We've talked about repurposing virus gene products, in particular retroviral gene products, on a number of occasions, and this is another example.
Now,
right. In Journal Club this morning, I was telling Akira Ono that we were going to do this paper, and he said, "Oh yes, the cell hijacks the virus." Then he said, "Wait, the cell hijacks the appropriate virus."
But these were here first, before retroviruses. And where they came from is probably another cell. And so it's a conundrum to know who's hijacking who.
I like that.
Yes.
Okay?
Right.
Because if a LTR retrotransposon predates viruses, it probably came from a cell and was very effective at moving around. Where did this GAG gene come from? Right? So I don't know. There's an interesting question. So the only repurposing here is that you've removed the GAG from the LTR retrotransposon. It's ended up in the genome by itself, and now it has a purpose, which is probably different from its original one. But this is very shrouded in mystery, I think.
So where does the envelope come from?
Oh, that's a good question. That's a good question.
As a late arrival, I missed a lot at the beginning.
There was no
there
written
was-
address on it. We don't know.
There's nothing before this.
Who
Dickson.
owns the sequence?
We're just starting.
Who owns the sequence to the envelope?
It's a good question. Because here's the thing, and this will make it even more complicated.
Oh, good.
If you remember, the fusion protein of gametes, tetrahymena, and what was the other one?
Euplodes.
No.
Paramecium.
Begins with C. It's got chlorophyll.
Chlamydomonas.
Chlamydomonas, right.
Chlamydomonas.
The gamete fusion protein seems to be related to viral fusion proteins. So was the cell fusion protein there first, or was the viral? We don't know. It's a good question. I love thinking about these things, but
Indeed.
this one, ARC is not taken from a virus. It's taken from a retrotransposon, which is a transposable element, and there are all sorts of transposable elements in many different cells. And I have a whole book here, a really thick book on my shelf called Mobile DNA. It's all about all the different kinds of transposable
I always think of
elements.
Barbara McClintock when I think of jumping
Sure.
genes.
So LTR retrotransposons are classified into different, I don't know what to call them, families. There's the TY1 copia-like ones. There are the T3 gypsy-like ones, and then the Bell PAO-like ones. Okay, so they're classified according to the sequence. And some of them are in pretty high copy number, up to a few million copies per haploid nucleus.
Good heavens.
In animals, fungi, protists, and plant genomes. And so they're all over the place. Retrotransposons, and they move around and they can do things. They can do good things and bad things. And that's really, I find that fascinating. And there are whole meetings just devoted to mobile DNA. They're very cool. So this
I taught a whole course on transposons shortly after I got to Buffalo, and it was that was one of the most fun courses I ever taught.
I'm sure it's probably a lot of fun to teach a course where you have to learn everything. Too, right? All right. So there was already some evidence that ARC had some gag-like properties. It's got some homology to gag proteins. And so the first, in this one paper on the mammalian arc, they show this, that you can see, they do phylogenomic analyses, and you can see orthologs of murine arc throughout the tetrapods. Do you know what a tetrapod is, Dickson?
That's a four-legged animal.
Yeah, because they have mammals, birds. Well, birds don't have 4 legs.
Tetrapod.
They have wings.
They have wings, though.
Mammals, reptiles.
If we did 4 of these, would be a tetrapod.
I guess the wings.
Right, the wings.
Tetra podcast. Yes, let's do that.
Just about mammals. Just about 4-legged mammals. That's all it is.
Coelacanth, zebrafish, carp genomes. And these are TY3 gypsy-like retrotransposons.
Gypsies.
All right.
They're genes without a home.
So one of the first experiments they do is they produce rat arc.
Right, so they had, you mentioned fish, but the conserved orthologs of ARC are in the tetrapods, but are absent from all the fish.
They're
except coelacanths,
closest relative,
which
Really?
are tetrapods, I think.
You might be right on that.
Well, the orthologs, but
the closest ARC in the coelacanth, zebrafish, and carp were
these gypsy retrotransposons.
yes, right,
Right.
the TY3 gypsy.
It's not really an ARC gene. It's an ARC homolog,
Right.
which in this particular case
Right.
is actually in the transposon.
Basically, this figure shows that tetrapod and fly arc genes originated independently from distinct lineages of TY3 gypsy retrotransposons, but they have good homology in the retroviral gag domain.
Right.
Mm-hmm. Right.
So this gene shows up independently, which is going to be even weirder when you get to the rest of the paper.
So the reason
these gypsy transposons are really, really old.
they're
Yes.
very old, yes. Now, they keep calling these retroviral gag domains because that's where it was first named in a retrovirus, but it was actually there in retrotransposons before that. All right, so the first experiment is very cool. They produce rat arc in bacteria, all right, and they purify it, and they look at it under the microscope, the electron microscope, and it makes particles that resemble virus-like capsids. Of course, when you produce gag from a retrovirus, they would make a capsid as well.
Right.
A double shell structure, 32 nanometers in diameter. And the same thing with Drosophila arc homolog, self-assembles into these virus-like structures. So that's pretty cool. It must have been exciting when they first did that.
Oh, wow.
In the retroviral gags, the C-terminal domain of the capsid protein is important for self-assembly, and they showed the same thing. If you remove that, they can't assemble into capsids. They produced ARC in mammalian cells, HEC293 cells. They don't have endogenous ARC, which is interesting. Must have lost it, I guess. And it also forms virus-like particles in these cells. Okay, so ARC makes something that looks like a virus-like particle, and that makes sense because it has some homology with GAG. Next cool thing is that this ARC will bind RNA and encapsulate it. Just like retrovirus, of course, the capsid picks up its RNA, and ARC does that as well.
What RNA?
Okay, so that's a great question. They produce ARC in bacteria. It picks up a very abundant bacterial mRNA called ASNA. But if they have ARC mRNA in there as well, because that's how they're producing the protein, that also gets incorporated. But if there's more ARC mRNA, then there's more in the virus-like particles. So it seems to be taken up. Whoever's there, it takes up. And is protected inside of these virus-like particles. So very interesting. In vivo, they immunoprecipitate ARC protein from mouse cortical lysates. They take a mouse brain and they lyse the cortex. They immunoprecipitate the ARC protein with an antibody. They then ask, is RNA in there? And the answer is yes. They can use PCR, reverse transcriptase PCR, to look for ARC mRNA. It's in there. So in vivo, neurons are binding ARC mRNA to ARC protein. This assembly requires RNA, interestingly. If they purify the ARC protein and get rid of all the nucleic acid. Doesn't make capsids. It doesn't associate with RNA. So that's interesting. And that's
which is common of capsids as well.
Yeah. If it's got RNA around, it's going to bind to it.
Right.
Now, the next question is, we haven't seen, I guess we've seen some kind of neuron in the mouse brain lysate, but now we're going to look at neurons in culture. So what they do is there's previous evidence that these proteins are released from cells, and this is something we didn't mention. I'm very sorry we didn't give you this background. ARC is thought to be involved in intercellular communication via extracellular vesicles.
Just like neurons.
Right.
Yeah, exactly.
Yes.
Microvesicles and exosomes, two different kinds that differ in size and origin. Okay, so now they want to know if this is in vesicles.
Right.
So they take media from mammalian cells that produce ARC because they put the DNA for ARC into it. They purify the extracellular vesicle fraction, and they can find ARC protein in it. And if they use a ARC DNA encoding an ARC that lacks the C-terminal domain, which we said was important for assembly into virus-like particles, there's very little RNA in the EV fraction, the extracellular vesicle fraction. So these vesicles are produced from cells, and they have the RNA in it. I also looked in cultured cortical mouse neurons. These produce vesicles, and they can find the ARC protein and the ARC mRNA in the extracellular vesicles produced from cultured mouse neurons. Yes, mouse neurons. And the protein is present in they stain it with immunogold. They can see the protein in these extracellular vesicles. And they do a cool experiment where they treat these cultured cortical neurons. They're cultured because they go to the opera.
Yes.
They're cultured, and they treat them with potassium chloride, which increases their neuronal activity. And you get more ARC in the media. Okay. So that's interesting. Higher neuronal activity, higher ARC being released. So that's pretty cool.
So these extra...
So getting the picture, Dickson?
Well, somewhat. The vesicles that are produced with the RNA inside go between neurons?
That's the idea. We haven't done that experiment yet, though, right?
All we know so far from the paper is that if you take mouse brain, you can find extracellular vesicles that have this stuff in it. Or if you culture mouse neurons, they will make extracellular vesicles that have ARC and ARC RNA in them. But we don't know yet about transfer from one neuron to another. It's implied based on everything else we know about ARC.
Well, if that's the case, then what do you think the RNA is doing after it's released in the next cell up?
Geez, you're about an hour and a half ahead of us.
Oh, good. Oh, while I was at Rockefeller, there was a group there working on the mechanism of action of black widow spider venom. And I think it has an effect on the formation of these vesicles.
It might.
I think someone should look that up as we're speaking, because
bad for you, right, Dickson?
That's terrible for you. Yeah, that's not good stuff.
In the next experiment, they take extracellular vesicles with ARC that are produced in cells. They take the media containing these, and they incubate them with fresh cells, mammalian cells. So now the question is, what you just asked, but there's not a neuron yet, but we'll get to it.
Right.
And it's just a plain old mammalian cell,
fibroblast or something.
Human embryonic kidney cells.
Okay.
If you take medium from ones that are producing ARC and put it on fresh cells, what happens? And the answer is that the mRNA gets into the recipient cell. It seems to be dependent on endocytosis because an inhibitor of that process blocks the uptake.
Okay.
And you can get transfer not just of ARC mRNA, but any RNA that's taken up into the vesicles, like GFP. If you have GFP in the originating cell, the mRNA will get into these vesicles and into these ARC particles and get out in extracellular vesicles, and you can add it to another cell. I don't know how efficient it is. You know,
Does the mRNA then produce a protein,
Yeah.
which is absolutely determined by the RNA?
yeah. So in this experiment, they looked.
Incidentally, just quickly, alpha latrotoxin, the black widow spider
Yes.
venom toxin, stimulates neurotransmitter release.
Release. Okay, the opposite.
Yes.
The opposite.
So
And-
go ahead.
Well, I was going to say, while we've digressed a bit, HEK, human embryonic kidney, 293 cells, or
Right.
as the adenovirologists just know of them as
293.
293 cells.
Yeah.
They are probably not kidney cells. They probably are an embryonic adrenal precursor cell, and those have many neuronal properties. So that might not have been the best cell to use as your non-neuronal cell type.
Huh.
That's what you were trying to do.
Yeah. Although they say they don't make ARC.
They don't make ARC. That my guess is that's what's important for them. They don't.
Yeah,
have the background.
So this spider venom would be interesting to know if you put that on neurons, if it make more ARC particles, right? That
Yeah.
would be interesting.
Either way, you can
Yeah.
still use it as a probe.
So the next experiment, they say, can ARC capsids transfer the mRNA into neurons? So they have cultured hippocampal neurons from mice knocked out for ARC. They lack the ARC gene. There's so many good things to say with this ARC, right?
Yes.
I'm waiting for Alan.
That's it.
And so they can transfer ARC mRNA into these neurons.
And by the way, in case anybody is wondering,
We are.
an ARC knockout mouse
Oh, yeah.
is viable.
Uh-huh.
Okay,
Uh-huh.
you get a mouse out the other end who seems to be able to take care of himself, but has numerous measurable cognitive and social deficits.
Wow. I bet it can.
Consistent with stuff like schizophrenia, if you want to make
And
that
probably no
extrapolation.
memory. It probably doesn't remember anything.
It has some memory deficits. Okay, but it can apparently do well enough to live a relatively normal life. Okay,
Yeah.
but
How does
Right.
it behave at the opera?
Yeah.
they
Exactly.
can't stand the opera.
Probably doesn't pay any attention. Doesn't have any focus.
That's right.
Very interesting gene. Wow.
Gee, what-
Anyway, the you can get uptake of these ARC capsids into neurons. And then they ask, well, what about ARC capsids made in neurons? Do they do the same thing? So they take ARC knockout cultured hippocampal neurons. They incubate them with purified vesicles that have or have not ARC capsids in them. And they clearly can take these up. So even ARC particles made in neurons can be taken up into other neurons as well, and endocytosis dependent. So we're getting this picture that, and these are natural ARC particles, not synthetic ones that they're making themselves, but they can be released from neurons and they can get into another neuron. So it makes you think about synaptic transmission, right?
Mhm.
So you build an arc, you load it up, and you send it on a voyage.
That's it. I'm waiting. That's what I was waiting for.
Yes.
I was waiting for something that was 2x2. So
Oh,
Yes.
2x2. All right, now the next part is about the transfer can undergo activity-dependent translation. Now, this is where it stretches the my abilities here, but let's try this. I got lots of help here.
I was just going to say you're on your own.
Oh,
Yeah.
okay,
exactly right.
but just tell me if I'm wrong. Okay. So the
I'll
idea
try.
here, I know just by being here, I'm wrong. If the mRNA is transferred into a neuronal cytoplasm, then they say we should observe an increase in dendritic ARC protein by inducing translation of mRNA through activation of a glutamate receptor. They have an agonist that will activate this receptor, and it should increase dendritic ARC. That's the one presynaptic is dendritic, right?
Axon and dendrite. Yes.
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 Presynaptic.
Pre.
Yes.
And they've shown this previously for endogenous ARC. If you treat with these GABA agonists, you get increase in ARC, right? So they're saying we should get more of these particles. And
What
so
about electrical stimulation?
They don't do that.
didn't do that.
Right.
I don't remember if they do it in the next paper or
Okay.
not.
Right.
No.
But that would be interesting to do that, right?
Sure.
Of course. Are you volunteering?
Absolutely. There's nothing left upstairs anyway. I've used up all my ARC.
So they treat what are they treating? Must be one of these cultured neurons.
I think they're
hippocampal, yeah.
cultures.
Anyway, so they treat them with the agonist of the GABA receptor, and the levels of ARC go up. And it depends on protein synthesis, as you would expect. So
Sure.
That's a cool experiment, because something that's affecting a known neurological, physiological receptor, GABA receptor, is affecting this. So basically, ARC capsids transfer the mRNA between neurons. The mRNA is available in the cytoplasm of dendrites for activity-dependent translation.
Yeah, we can come back to this later, because there are some differences between these papers.
Oh, yeah, for sure.
Okay. And we got to talk. I at least want to talk about dendrites and synaptic boutons later on.
Okay.
I don't.
We could talk about it now, but I think we need to talk about synaptic boutons and the other paper first.
So the other paper is from, as I said, University of Massachusetts in Worcester. And this one...
Worcester.
This one is also about ARC, but from Drosophila. And there we have DARC, which reminded me of Jeanne d'Arc, of course.
Yes.
D-A-R-C.
So D for Drosophila.
Drosophila.
Right.
So you can find, it's been known for a while that DARC1 is in Drosophila, and cultured cells. What they found in this paper is that cells from Drosophila produce extracellular vesicles that have DARC1 protein in it, in these similar particles. They actually take, so Drosophila S2 cells, it's a famous cell line from Drosophila. They take the supernatant, which has the extracellular vesicles. They purify them by sucrose differential centrifugation and sucrose gradient sedimentation.
Dun, dun, dun.
Sucrose gradient.
Great old stuff.
Old stuff.
Sweet.
They take the extracellular vesicle fraction of the gradient. They treat it with RNase. Okay, and so if anything's in it, it's protected. And then this is great. Never see this anymore. They extract poly-A plus RNA, and they make libraries of DNA with reverse transcription, and then they sequence that. They say that about 100 transcripts were enriched in this fraction, at least twofold, including the most abundant was DARC1 mRNA. DARC1. Okay. And they show that this mRNA is present in the larval Drosophila by doing in situ hybridization. And an antibody against DARC1 is also shows that the protein is there as well. And they show that this is in the presynaptic compartment, this DARC1 protein. Presynaptic, present in a punctate pattern.
Yeah, so maybe this is an opportunity to talk about the structure of a neuron.
Go ahead.
I'll try. As a matter of fact, I linked in, if anybody watched it, this little 2-minute video on the structure of a neuron that might well go into the show notes.
Hey, before you do that, let me just make a correction.
Okay.
The experiment is important because here they're looking at the neuromuscular junction.
Right.
Okay. So they wanted to know whether DARC1 mRNA is at the neuromuscular junction. So they did hybridization. They also did immunohistochemistry with an antibody, and they find it's in the presynaptic compartment. It's in the nerve, then there's the synapse, and then there's the muscle. Okay. So I didn't.
They're very distinctive junctions between neurons and muscles.
Right. Okay. Rich, go ahead.
So picture a cell, a blob with a nucleus, okay? And just for the sake of illustration, put a bunch of finger-like projections out over to the left, okay? And
you're right.
And my left is I'm looking at the universe, okay?
Right.
And call those dendrites. And then out the other side, put a big, long, thicker projection and call that an axon.
Exactly.
Okay? And what happens in signal transduction is that a signal comes into the neuron on the left through the dendrites, okay, and is transmitted through the cell body, where if conditions are right and there's enough stimulation, it transmits a signal down the axon to the right and out to whatever that axon may be contacting. It may be contacting another neuron through that neuron's dendrites. In the case of this Drosophila thing, it may be contacting a target tissue such as a muscle.
Right.
At the end of the very tip of the axons are these synaptic boutons, French for button. They're these little bulbous things that come off the end of the axon that are important they actually are important in making the contacts in something like a neuromuscular junction, and I suppose in regular nerve transmission, though I don't know enough to verify that. So dendrites and axons are receiving the input signal, propagating the exit signal, respectively.
Right.
Got it. All right. Now, I misspoke. They're looking at the Drosophila larval neuromuscular junction. They find dark 1 mRNA and protein at both the pre- and postsynaptic
Both.
part. Yes, both. mRNA is enriched in these extracellular vesicles, and both mRNA and protein are present in the pre- and postsynaptic sites of the neuromuscular junction.
Right. And correct me if I'm wrong, but what I get from that is we're looking at where the axon contacts the muscle.
Correct.
Yes.
And talking about the presynaptic part of that means we're talking about the neuron, the tip of the neuron. That's the presynapse.
Right.
Postsynapse, we're talking about the muscle tissue.
That's right.
Okay. So there's ARC on both sides of that junction, on the nerve side and on the muscle side.
But the arrangement of the contact point is unique. It's got it's kind of a elliptical oval type of structure that it actually puts up against the muscle cell to depolarize the muscle cell to allow calcium to flow into the sarcoplasmic reticulum and therefore allow the actin and myosin filaments to do their thing.
Awesome.
Because trichinella does something along those lines.
Huh. Right now they say, okay, this dark 1, it's got a part of the open reading frame looks like a gag protein.
Oh, come on.
So they're saying
can't be everywhere.
Maybe these could make particles that would transfer the dark mRNA across the synapse. So
right. Now, this is the same finding discussed in the previous paper where they noticed the ARC has this gag-like sequence.
Right.
The Drosophila ARC, dark, has this gag-like sequence.
So now they're going to test it in their Drosophila. They downregulate dark 1 in motor neurons specifically. All right. And they use a particular construct which will deliver RNAi to motor neurons specifically, probably a promoter, right, that does that. And then they look at ARC pre- and post-synaptically. All right, remember, they're doing this in neurons, not in the muscle. Right. They're knocking down ARC. So using this RNAi in motor neurons, they get a decrease in dark 1 mRNA by in situ hybridization in the presynaptic bouton and in the muscle postsynaptic region, even though they are only knocking it down in the nerve.
Right.
So it also goes down in the muscle. So of course that implies that it's flowing across, right, in some way, and therefore you hit the source and you knock down what's in the muscle.
Yeah.
And so they do quite a few controls to show that this is correct, but they end up saying probably most of the postsynaptic dark mRNA comes from a presynaptic pool. And they think, well, it must be extracellular vesicles, right?
So the neuron is pumping dark to the muscle.
Correct.
Dark mRNA.
So one of the things I really like about this paper is that this is all in effect in vivo. We're talking about fly larvae.
Fly larvae, yep.
As opposed to, I'm not knocking what's going on before because, flies have advantages for doing all this kind of stuff, the genetics, the ability to manipulate the embryos and everything else. But in the previous paper, we were talking about predominantly cultured cells. But here we're talking about real larvae where we can go through and look at these synaptic junctions as they exist in the larva.
Right. They did another cool experiment where they produce DARK1 in the muscle, but it doesn't go back the other way.
Right.
So, I'm sorry, they produce the RNAi in the muscle, and it doesn't affect
they can't inhibit
what's happening in the presynaptic.
In the nerve,
so it's not going the opposite way.
Right.
And then they do an interesting experiment where they use a dark transgene, which they produce in neurons lacking the 3' untranslated region of the mRNA. So again, you do the experiment where you put this in the nerve, the motor neuron, and then you see if it's in the muscle. If you take off the 3' non-coding region of the mRNA, it does not transfer into the muscle.
And they had a lead here because typically there is a packaging sequence for RNA into these gag particles that is typically in the 3' UTR, and they I think they even could identify a bit of a sequence. So they aren't just pulling that out of thin air.
That's right.
So I'm going to ask again, can you come back out of the weeds and tell us what the 3' UTR experiment tells us?
Out of the weeds?
I think we're lost in the detail weeds here.
So the 3' non-coding region of the mRNA between the stop codon and the poly A tail, if you remove that from this dark construct, from this dark gene, it's no longer packaged into... packaged. The assumption is no longer packaged into these virus-like particles, but all we know here is it doesn't get from the nerve to the muscle across the synapse without the 3' UTR.
The implication is that there's a very specific packaging process that involves recognition of the RNA specifically by the gag protein to package this RNA into the dark particles.
Right. It's not just packaging up any old RNA and selling it.
No.
And the 3' UTR is part of the specificity.
Yes,
Yeah.
That's part of the tag.
That's what I was trying to get. Okay.
Yeah.
Yes.
And I think they do an experiment that if you just have the 3' UTR alone, that can allow transfer without any...
they hook it up to GFP, right?
Yes. So you can, if you... 3' UTR on GFP, and you have to make dark, of course, in some way, DARK1 vesicles, but that'll pull GFP over, just the 3' UTR. So that's got the signal in itself. You know, the dark mRNA encodes the capsid, and then that capsid wraps up the dark mRNA via the 3' UTR, and that wrapping up can be conferred to anything else. So you could deliver things across a synapse this way, whatever you want, right?
Now, here's the difference between this paper and the previous paper, okay? And that the previous paper suggested that the packaging was not necessarily specific. They got packaging of RNA more or less in proportion to the abundance of the RNA. So I think that this probably needs to be resolved, but we're talking about the difference. We're talking about flies versus mammals to start with. So there may actually be a difference, but we're also talking about the difference between these in vivo experiments on the one hand and more or less in vitro experiments on the other hand.
That's right.
And not only is this flies versus mammals, it's flies versus mammals of a gene that we know it essentially evolved independently in these two lineages.
Right, exactly. So there could be a real difference, or it could be an experimental system difference that'll be resolved down the road.
Right.
It's clear in this case that you're specifically packaging it's clear to me anyway, specifically packaging the dark RNA based on its interaction with this sequence on the 3' end.
The one question I have in my mind is to think about a normal nerve communicating with its target, be it another nerve cell or a muscle cell. It uses neurotransmitter substances for that, right?
Yeah.
And this is not a neurotransmitter substance that it's packaging up. It's packaging up an RNA. So what do the vesicles look like that contain the neurotransmitters? Are they also ARC?
I don't know. It's a good question. I don't think they looked at that. You know, whether there's neurotransmitters in the same vesicles or not. Yeah. I don't know. Don't know. I don't think they.
My impression has always been that neurotransmitters are just are in membrane, soluble inside a vesicle.
And that's
But
what I
maybe
asked. Yeah, that's exactly what I asked. What is the
That's
vesicle
always been
made
my
of?
impression, but that's
me
before
too.
I knew this, you know.
Well, what
Right.
is the vesicle then? Is it another arc molecule? That's all.
Or is it just a vesicle?
My guess is that everybody's going to go looking.
Yeah, but it's got to be made out of something.
So the next thing they do is they follow up on the retrotransposon connection, which they haven't done yet in this paper. But they find a common Drosophila retrotransposon called copia is the mRNA of that is enriched in these extracellular vesicles that they purify from these Drosophila cultured cells. And they show
There are
that
copious amounts of
they're copious. That's copia. And this mRNA encodes mainly the gag region. And that is the connection with the previous paper. It's a gag from a retrotransposon that gets incorporated into these particles, that makes these particles, and the mRNA gets incorporated into it. They show that this protein, this dark one protein, binds its own mRNA. They do mass spectrometry analysis of extracellular vesicles compared to total cell proteins, and the dark proteins are enriched there. So the idea is that that this retrotransposon gag is making these particles, and that's what's transferring across these synapses from nerve to muscle. Yeah.
So what would happen if you had two kinds of cultures? You had unmixed. You had one set of cells which has obviously the vesicle and the arc and the arc I meant to say and the messenger RNA, and then the other cell that it's communicating with doesn't. Would you eventually find versions of the reverse transcriptase DNA in the nucleus of the arcless?
No, that's a good question.
Would dark copy itself into the other cell?
Correct.
So you would need reverse transcriptase to do that. They actually mentioned that in the discussion that these retrotransposons are not known to go from cell to cell. They mainly jump around, but why not? I mean, if cells are shedding vesicles all the time, why not, right? That must be how it got around initially.
Exactly.
Right?
Exactly.
And could the reverse transcriptase be provided in trans?
Exact.
I don't see why not.
So if you've got one of them in the cell, then you could get any of them.
Because the mRNA that's present in these vesicles doesn't have RT. It's just gag, right?
Right.
But maybe.
But if it gets in and there's RT floating around,
of course, there might already be RT in the recipient, right?
Right. So, that's what I mean.
Yeah, could be.
I mean, this sounds very self-serving, doesn't it? This sounds very selfish.
Of whom?
Of these vesicles and the RNA. It just sounds like I want to make more of myself. It's almost like a virus.
Well, they're sometimes called selfish genes, right?
Almost like a virus. Yeah.
Well, so
what does it do? What is it doing there?
Which one?
Getting around.
Yeah, but that's nice. But what is it doing?
When we Dickson, when we get down to the bottom line here,
we're almost there.
We're not going to know what they actually do.
That's very unsatisfying.
I'm sorry.
So
I should have come in later.
They go ahead and show that this ARC protein will bind RNA. It will form capsid-like structures. They produce in bacteria. They look under EM, just like the previous paper, except we're using the Drosophila version. They can see 40 nanometer round structures. Okay. And then they do some synaptic plasticity experiments. Okay. So the neuromuscular junction expansion, which occurs during larval development, it involves addition of new synaptic boutons. Okay. So they want to know what's happening with dark during this period. Okay. And here you can look at larval development where there's differences in the synaptic plasticity. Okay. So in the third instar, they see a reduction in bouton numbers just before metamorphosis in the null mutant of dark compared to controls. Okay. And you get the same effect if you produce an RNAi for dark in motor neurons. You get a reduction in bouton numbers if you remove dark during larval development. And they say this suggests that reducing dark in motor neurons inhibits synaptic expansion, which usually occurs during larval development.
Right.
And this can be rescued by presynaptic production of dark 1. So you need dark apparently for this synaptic expansion. This expansion involves formation of immature boutons, which don't have neurotransmitters. Okay, and mutations in dark or downregulation of dark neurons result in accumulation of what they call ghost boutons. These are kind of immature boutons that will eventually mature into the real thing. And again, this is rescued by expressing the wild type gene.
What we don't know is how.
We don't know how. We just know that there's a correlation between bouton number and dark one. Right. They also say, and I take their word, neuromuscular junction expansion can be stimulated by increased synaptic activity during larval development. And so they reduce dark by RNAi, and then they look at the formation of ghost boutons upon spaced stimulation, acute spaced stimulation of motor neurons. So this is, I'm sorry, I don't know what that means.
It means electrical stimulation.
I think they're jolting the flies. That's my guess. Yep.
So space stimulation gives rise to more synaptic activity, and this goes down if you don't have dark, basically. So you need dark for developmental synaptic plasticity and also acute plasticity in response to electrical stimulation. Mm-hmm. And that's basically it. Well, there's a little more, but we don't need to know.
So, Dickson, I've been reading here about synaptic vesicles.
Good.
I don't think they're going to have these things in them. Okay. First of all, these dark particles or the arc particles, whether they're from mammals or flies, are on the order of 30 nanometers.
They're small.
Diameter.
I agree. They're small.
Well, they're still synaptic vesicles themselves are only about 40 nanometers in diameter. Oh. Okay. So you would only be able to get like one of these in each synaptic vesicle. And in terms of the protein composition, they've got the protein composition pretty much figured out. There's transport proteins like proton pumps and trafficking proteins that are membrane proteins of one sort or another. So I think that synaptic vesicles are going to be quite a different beast than these things.
Thank goodness. So these arc vesicles probably don't interfere with neurotransmission then?
Probably not.
Because they're not similar in terms of...
They certainly seem to be important for neuronal development.
Mm-hmm.
Right.
Synaptic plasticity.
On the other hand, there's this bit right at the end where they apparently previous work had found that dark wasn't involved in plasticity.
Right.
And they eventually chalk this up to strain differences.
Right.
There's another strain of flies where dark levels are very low. And so it's weird because this should be necessary, and yet just in a different strain of flies, it's at a low level. And I'm wondering if maybe there's a different protein that serves the same purpose in the other strain.
Must be, right?
Right. And if this arose multiple times in evolution, then maybe this is something that you can pick up in a variety of ways from different retrotransposons.
Yeah. So here's the bottom line. Here is the executive elevator summary. We have ARC in mammals and flies, which is a gene that is incredibly important for neuronal function, particularly at synapses between neurons or neuromuscular junctions. And signals are transmitted across this junction by packets of a protein that forms a capsid, and this contains RNA. The RNA is then delivered across the junction, and what it does, we don't know. But the cool part for us on TWiV is that these virus-like particles are made up of a gag protein, which is probably a precursor to a retroviral capsid protein. I think the most interesting question is what came first, and I said this before, what came first, the gag or the virus? And we think the gag came first. And why? What was it doing? I can't ask why, but what was the function of that gag originally? Maybe this arose. Hey, get this. Maybe gag arose as a neurotransmitter, like we've been describing here, and then it was stolen by viruses.
Right. Could be.
Or it evolved into viruses.
Yeah. Of course, that would be the way to do it because virus couldn't steal it if they didn't exist yet. But
yeah, or going back further, not even as specifically a neurotransmitter. Maybe this was a cell-to-cell communication tool.
Sure.
Because you can transfer RNA to another of your siblings or whatever.
Wait a minute.
And then a virus comes along, or this turns into a virus, and things get out of control.
Can you gag?
So there are transposons that are just DNA and a reverse transcriptase, right?
That's correct.
Not all transposons necessarily have a capsid protein.
Correct.
But if you are a sly transposon that doesn't have a capsid protein, you might think, aha, I could get around better if I could package myself and get to another cell. I'm going to steal this neurological protein and do it that way.
Rich, you make a great virus.
Okay?
By the way,
after 45, 50 years, I've learned how to think like a virus. That's important.
How many genomes? No, forget it. You can gag me with a maggot.
Ooh.
Yes.
That's the name of our issue.
This makes me think.
Gag me with a maggot.
This makes me think, and we'll talk about this maybe next time. So we talk about how the placental syncytians were exapted from retroviruses, and those are envelope proteins. So they have to be exapted from retroviruses and not retrotransposons, because retrotransposons do not have envelope protein.
Bingo.
Got it. Thank you. You like when you...
I do. I like that. Like you're divvying these things out into nature according to Vincent.
And so for the people who are handing out the money out there, the politicians, we're talking about work that has relevance to mental health, schizophrenia, Alzheimer's, autism, okay? And some of the most important findings are coming out of fruit flies, dudes.
Yes.
Fruit flies.
And the original retroviruses that were studied to give, insight into gag proteins, avian retroviruses, mouse retroviruses, way before HIV came on the scene.
Right.
All right. So that fundamental research, got to do it. All of this wouldn't make any sense without that. Very cool stuff.
Today I had the occasion of addressing 9 undergraduates at Fordham University for the first day of my class, and I showed them a genome analysis of life on Earth. These big circles with all kinds of small lines and smaller lines and smaller.
You mean a pie chart?
It's not a pie chart. It's like it's bigger than that. It's got all the animals and plants on it.
Cake chart?
Cake. It's a multi-layered cake chart. And I started with telling them about the genome of bacteria and how simple that is. And then how life, if you start looking at it, it builds upon that simplicity into complexity and more complexity and more. And this is absolutely stunning.
But complexity is not necessarily better.
No, it's not.
Because the orchid genome is bigger than ours, right?
No, but that's polyploid.
Although you could say orchids are nicer than we are.
It's polyploid.
Dickson, what's the course?
It's called Ecology for Designers.
Oh.
And there's a lot of crosstalk between designing and nature. And, I like to show them the connections between how, let's say, a hardwood forest functions during a drought or during a flood, and then how we manage the same thing and how nature communicates much better than we do. So
you talk about biological mimicry?
I do, a lot.
Very good.
A lot of that.
All right.
But they got it. They actually got it.
Let's do a couple of emails.
Oh, before we leave the pie charts, I just wanted to insert that in Brazil, they call them pizza charts. And in France, they call them le camembert chart.
It's a living pie chart.
Diagram and fromage, cheese, or sometimes a cheese diagram. So it's funny that they're always some kind of food.
Exactly.
Yeah, people are hungry.
That's it. It's got to be right.
Don't we like food?
We do.
Yeah.
We do too. Alan, can you take first email?
Sure. Daniel writes, hello to the esteemed TWiVarium. I had recently seen a TED Talk in which the speaker, Professor Goodkoff from Roswell Park Cancer Institute, puts forward 2 ideas. To me, both ideas sound intriguing and quite radical. My limited knowledge of the virus canon is expanding with each new episode of TWiV, and as I go back in the catalog of older episodes you frequently reference in your show, but is inadequate to critically evaluate his proposals. I was compelled to write to you for comments and dissections of these ideas. Here, to the best of my understanding, are summarized ideas from Dr. Goodkoff. First, viruses are responsible for diversity of mammals in evolutionary history. Second, viral sequences that integrated in the genome were over time silenced by mammalian cells. And in recent times, these integrated sequences determine the age of cells, acting as clocks and becoming awake in aging cells, resulting in senescence and leading to cell death. Have you come across research supporting either of the two hypotheses, and what would you say about them? Also, I'd like to wish you happy holidays and thank you for great content provided by TWiV and the TWiX podcasts. I anticipate new episodes every week and look for time in my schedule to fill with TWiX episodes from the back catalog. Thanks again to Vincent and all the contributors.
Right.
All right. So viruses are responsible for diversity of mammals in evolutionary history. I don't know how strongly the claim was made in the TED Talk.
But if they are the only cause, then that would be wrong, right?
I wouldn't say, I mean, diversity in mammalian history has been driven by the same things that drive speciation. See Darwin.
Correct.
Yeah.
You know, this goes back to bacteria and before, right?
Sure. Random
but a virus is playing a role in driving the diversity of mammals? Absolutely.
For sure. I think responsible is the wrong word, right?
Yeah.
Because there are a lot of factors.
So these can be a powerful selective pressure. They can be a powerful pressure for speciation. So gene exchange, right? Bringing in new genes. Well, that's right.
Yeah, for sure.
So a weaker version of that than
or killing off populations.
That too
is totally plausible.
Yeah.
Yes.
Yeah.
Second, viral sequences that integrated in the genome or over time silenced by mammalian cells. Sure.
Yep.
See transposons. And in present times, these integrated sequences determine the age of cells acting as clocks and resulting in senescence and leading to cell death. I'm not sure what the reference is here.
I've never heard of that. I think it sounds to me like you have an endogenous retrovirus, which is silenced, and then it awakens at some point and kills you. That's not what happens, for sure.
Right.
I don't know.
I mean, there may be components of the apoptotic machinery that are borrowed from old retroviruses. I don't know.
Scientist and expert tells us what aging really is, why it happens, and what our future might be. No, I mean, it's not just reactivation of silenced viruses. Aging involves other things like telomere length, right? So,
and oxidants, not accidents, oxidants.
I don't think that is what leads to cell death, awakening of silent copies of viral genomes. But
so I don't know. I don't know what the argument is in the original TED Talk.
Okay. Someone look at it and tell us.
Yeah,
it's only 18 minutes.
I got it queued up here. Unfortunately, I haven't had time to look at it yet, but I will.
All right, we'll look at it next week.
We'll look at it
next week. We'll tell you for sure.
Right.
Dickson.
Dave writes, Virus Mavens, you might find this podcast interesting. And then he gives a listing of samharris.org/podcast/item/humanity.
Okay, you don't have to do all this.
Also, but it's a short, come on. Also, I must further the recommendation of Vincent's for Lane's The Vital Question, being a big Lane fan, starting with oxygen, the molecule that made the world. Weather here in Bend, Oregon, Rich, you should know about this. It's fair, 34 degrees F.
That's right. That's where Rich goes.
1 degree C. Humidity 75%. Wind speed 6 miles per hour out of the southeast. Barometer 30.1 inches. 2.27 F. -3 C. Visibility 9 miles. Wind chill 29 F. -2 C. Last update 01 December 9:15 a.m. PST, Pacific Standard Time.
Okay.
This is a link to an interview with Jennifer Doudna. Yes.
Probably pretty good.
Sure. Jennifer is very good. And the vital question is great. Oxygen, the molecule that made the world. I'm reading it right now. It's great.
Take a deep breath.
Kathy, can you take the next one, please?
Mm-hmm. Luca writes, Dear TWiV, TWIM, TWIP team, I'm a second-year biological sciences undergrad at the University of Birmingham in the UK. It is 4 degrees centigrade here. Firstly, I'm currently searching for a placement year opportunity for the third year of my course. I've applied to some places in the UK, like the Crick Institute and others, but I wondered if there are some lab-based and research-based placement year opportunities that you know of through your work. I've attached my CV if you are interested. Secondly, a big fan of TWiV, TWiP, and TWiM, and I've learned a lot from learned a lot from the show. I listen to it mostly on trains. The only thing I feel could be improved is the overall length time. I feel that 1 hour, 50 minutes, talking more about TWiV here, is too long for some listeners and tests their attention span. I appreciate that there are some things like the sponsors and discussion of papers and news that can't be cut down on, but I feel like the introduction of everyone takes too long considering most of the hosts are on each week. The new papers and stories are the real essence of the show, and sometimes I just feel the tangents go a bit too long, and I find myself skipping through some bits of the show. Also, just a small note that on some of the TWiP episodes, a host refers to a parasitic worm and measures them with a unit of length called the sonometer, trying to spell it phonetically. I Googled this word and a few spelling variants, but I couldn't find anything to do with length. Have I got the spelling completely off? Really appreciate the work you all do to promote science. Thanks in advance. Okay, so I have actually had someone ask me that same question, and so you guys were unfairly laughing at this person because, as I said, someone else has asked me that, and it is just a highfalutin way of pronouncing centimeter.
Indeed.
That's what they do in Canada, sonometers.
Yeah.
Is it Canada or UK?
Well, my wife's
my
wife's my mother-in-law was from Canada. She used to say sonometer.
I think it was just a doctor way of.
Yeah, she was a nurse. Maybe that was it. Yeah.
Yeah.
I mean, we don't mean to laugh at you. It's just funny.
We're laughing at the pronunciation of centimeter as sonometer. Which who on TWiP is doing that?
I guess it's
Daniel.
Probably Daniel. Got to be Daniel.
Yeah.
Luca, you found the solution to the long shows. Just skip through it.
Skip.
Yeah.
That's right.
Well, I would also like to refer Luca to TWiV 186, titled "From Buta to Stump Grinder."
Yes,
where we discuss this very topic.
Yes, we do.
You can also,
and I don't think she has to listen too far into it. I think we do it in the beginning of the show. Sorry, Kathy. Go ahead.
Yeah, no, I was going to say you can listen at faster speed. You don't have to listen to one episode per train ride. You can put it over 2 train rides or whatever. Yeah, we try as we might. We don't manage to shorten these much.
And it used to be a 90-minute show, so there is some fairness in the criticism that it has bloated.
Yeah.
I don't think the introductions take very long, do they?
I think actually the email and discussion around picks is what seems to eat up the most time. Like we're at an hour and a half now, and we've done the papers, we've done some emails.
Last week it was 37 minutes before we got to the paper because someone was pointing out to me that they didn't know what the paper was yet because they'd only listened up to 37 minutes.
We're going to get another letter. We're going to get another letter.
Question about placement year opportunities.
Oh, yes.
We don't have something quite like that. You would have to probably write to bunches of people on your own and, be willing to perhaps not get any pay for it, but just go volunteer somewhere if you wanted to do it elsewhere. So we're, at least in the U.S., we don't have those kinds of opportunities. We have summer opportunities, but not so much full years.
All right. Let me read the next two. Neil writes, "Hi, TWiV crew. Came across this article. Thought you and the TWiV listeners might like it as much as I did." This is an article from 538. There's no such thing as sound science. The easiest way to undermine good science is to demand that it be made sound.
This is a really good essay. Calls to base public policy on, quote, sound silence seem unassailable if you don't know the term's history. The phrase was adopted by the tobacco industry in the 90s to counteract mounting evidence linking secondhand smoke to cancer. And then science is not a magic wand that turns everything it touches into truth. Instead, it's a process of uncertainty reduction, much like a game of 20 questions. So the tobacco industry's tactic was to turn this baked-in uncertainty against the scientific enterprise itself.
Right.
And it goes on with most scientific controversies aren't about science at all, and once the sides are drawn, more data is unlikely to bring opponents into agreement. And then they talk about the difference between how decisions are guided in Europe by a precautionary principle versus in the U.S., where agencies tend to wait for strong evidence of harm before issuing regulations. Anyway, it's a really good essay, so I agree with Neil.
All right. And then one from Johnny. Greetings of the season and Boston's first snow. I think that was last year, probably. Certain I don't understand how they did it, but not bad art for DNA. Maybe a better flu vaccine. Tidings of comfort and joy. And it was last year, yes.
Had to be.
The biggest DNA origami structures yet. 3 new strategies for using DNA to generate large self-assembling shapes, create everything from a nanoscale teddy bear to a nanoscale Mona Lisa.
I looked at the pictures and I read the article, and then when I went back to the beginning, it said 3 new strategies, and I realized that I'd missed the whole content about the way the author had described the 3 strategies. So somehow the science writing itself didn't grab me, but the pictures were great.
Yeah. Great structures, for sure. All right. And then the last quick one, Anthony writes, female professors get more grief from students, study finds. This is in British Columbia. The article here, published January of this year, January 4th. Research has found that women working in education are more often requested to give extensions, boost grades, and be more lenient when it comes to classroom policy. What do you think about that, Kathy? Do you find that?
It's just the first I've heard about it capsulized in this way. But if there's data behind this, I would be really interested in that. I didn't have time to look at this yet. It wouldn't surprise me that they say gender could play a role in, how students interact with faculty. That's no surprise.
So the person who did this study said that we believe it's because women in general are expected to be more empathetic, more nurturing, more likely to be helpers, to assume a nurturing role. Well, I don't know how broad this study is, but, might be you want to do it in other places. All I know is people ask me for an awful lot of stuff in my class.
Yeah.
I get a lot of grief, but maybe women get more grief. All right, one more. And Rich, I think you haven't read one, right?
Yeah, I haven't read an email. That's right.
Go ahead, Max.
Some follow-up. Max writes, hello, TWiV gang. That's small g, capital A, small n, capital G. Looks like a antigen of some sort. I'm finishing my PhD in molecular biology. I study ribosome biogenesis. Good for you. And I'm hoping to do a postdoc in a lab that studies viruses. My interest in viruses is how I found your podcast, and both the science and discussion about science have been great. On to my comment. In TWiV 472 about the skinks, one of you, I think Alan or Rich, mentioned that it would be difficult for birds to fly whilst pregnant. That was me. It just so happens that there has been a great paper published on this very subject. I'll quote from the press release. Stoddard and her colleagues wrote a computer program. Oh, my goodness. Extractor. EGGXTRACTOA, or egg tractor, if you like.
I think it's egg extractor.
Egg extractor, okay. That picks out the egg in any image and measures its length, width, and shape. They found that good flyers tend to lay eggs that are more elongated and more asymmetrical, likely because lots of time in the air requires lightweight, compact bodies. Another random, long-standing science mystery solved.
So the eggs of better flyers are more aerodynamic.
Yeah, amazing.
Even though they're inside the bird.
Right, but they would have less of an effect on the shape of the bird.
Max is from Jupiter, Florida, where it was currently 21°C, partly cloudy, 8 kilometers per hour wind, 96% humidity. Sure is hard to complain about Florida winter. I'm with you on that. Stay warm. He says, P.S. Pregnant bats can also fly, but it seems like it's a real drag. And he's got a
great book excerpt about this. That bats apparently. Only one ovary is active. The other horn of the uterus is reduced. They say flight increase may limit the increase in mass during pregnancy because the additional mass basically increases their stall speed, increases the wing loading. So they have to fly faster to stay aloft, and it may have an energy penalty for them.
Amazing.
Yes.
Cool. Interesting.
Thanks, Max.
Thank you.
All right, let's do some picks of the week. Dickson, what have you got for us today?
It was today's article in the New York Times that said last year was the hottest year ever. Recorded.
Did you send that to
One of the hottest on record.
I didn't. I did not. It says hottest year.
One of the hottest years because last year was hotter when you click through, but they were surprised that the decrease to this year wasn't a bigger decrease than it was.
You mean
Right.
2016 was the hottest?
Right. 2016 was the hottest. 2017 was nearly as hot, and that's surprising because we didn't have a strong El Nino.
Okay.
So that's kind of disturbing. And they also had this very nicely done graph showing global average surface temperature from 19th century to now, and of course showing what everybody should be able to acknowledge at this point.
You know, it's incredible how fast all this has happened.
I know.
Yeah.
Because this is basically correlates with the use of fossil fuels,
Yep.
right? In particular for internal combustion
No
engines.
question.
Yes.
And I suppose also coal for heating to some extent. But mostly engines. And I mean, we're just talking about not even 100 years, right?
Yeah.
Right.
That's right.
Right.
Yeah,
Or, well, a little over
really takes
100
off.
years.
Really takes off in the 50s.
Don't forget about methane too, though. That's a big player. in
Sure.
like from cows?
Yep. And termites.
Really? Termites too?
Huge.
Sure.
From wooden houses?
There's a belt of termites across Africa and South America. It's incredible. It's just an amazing
Have
amount
we
of
have somehow participated in their expansion?
No, not at all. In fact,
All right.
we're
No,
trying to
what's driving this increase is human
It's
fossil
mostly-
fuel use,
mostly
principally,
If-
and human methane production as well.
and cattle too.
Right, that's human related.
That's correct.
Well, people who don't believe this, they say, "Oh, we need more studies to
Believe
go this."
crap on
but
that.
this is just
Again, back to my classroom, I used the example of prediction, and I said science is prediction, and I said, if it says 0 chance of rain, you're not going to take your umbrella. But if they say 95% chance of rain, you'll always take your umbrella. So there's a 95% chance we did this. So if they said 5% chance, and who cares? But 95%? Come on.
yeah.
It's not a belief.
What blows my mind is that the activity that's causing this, started what? They've got this graph starting in 1880, but
Yeah. They've got lots more data.
19th century, call it 1850, but I mean, really, the activity starts to ramp up at the turn of the century, and the temperatures are now look like they really kind of exceed the norm by about 1940. So we're only talking about somewhere between 50 and 100 years of this kind of activity to cause the damage.
Correct.
I mean, it's amazing how quickly.
Yeah, that's right. The other piece of depressing news was that recently, I forget which newspaper I was reading, but it said that the oxygen content of the ocean is dropping,
Yes.
Mm-hmm.
which means because it's getting warmer.
Yeah.
And that's all related. It's all depressing.
And yet we are rolling back environmental regulations that would
Yeah,
try and check this,
that's
right?
correct. That is correct.
And because
So-
we'd like companies to make more money.
It's criminal to deny this. Criminal.
Well, we should bring a suit against the
That's what
Yeah.
that's what
Good
Bill
luck
Nye
with
the
that.
Science Guy said, and that's also
Yeah, I
what Neil deGrasse Tyson said. It's criminal to deny this.
Yes. Yeah, good luck with that, right? We need to just change administrations and go back to regulating this stuff. Can we do that? That's doable.
We can do that,
That's doable.
and we will.
Alan, what do you have?
I have a paper that came out in Science. I guess this was this week. It was January 10th. Science Advances. So this is open access. This is Science's open access journal. Experimenter gender and replicability in science. And this is really, really interesting. This is a bunch of studies that I wasn't aware of because I don't really follow this field that much, but tests where they set up psychological or neurological type or physical type tests, and you have the test done by a Confederate who's, says that they're the researcher, and depending on whether that person is male or female, you can get different results. If you have, for example, if you're administering an intelligence test, people will tend to overperform if they have a male giving them the test and underperform if they have a female giving them the test. And it applies for both men and women. Tests of asking about pain. Some small pain is administered, and how would you rate this pain on a scale of 1 to 10? Men in particular will rate that as being lower if a woman is giving the test.
Interesting.
Because they want to be, presumably, this is part of the sexual dynamic between men and women.
Yeah.
You want to be tough, right? So they summarize a whole large collection of these studies and talk about the impact of this, the likely impact of this on various branches of science, including a lot of biomedical research. So you can easily imagine that if you're doing a study to look at efficacy of a pain drug, you get different results depending on whether you have male or female experimenters administering the test. How much pain are you feeling? And you could get one study showing that this is a highly effective drug and another study showing that it's a worthless drug. And then you'd say, well, these are not reproducible results. Why not? It may be that you had a gender bias in the result because the subjects are giving different feedback.
Amazing.
So the solution is we need to have androgynous robots doing the questioning.
That is one possibility. The other possibility is that you deliberately account for this in the study. And one of the things they point out is that very few studies say who did what, who administered the test, when in fact it could be making a huge difference in the result.
Yeah, maybe you could do a study with both genders questioning and see if it makes a difference,
so
right? Yeah.
You'd have two arms of the study, or you'd randomize people depending on, to different groups.
Yeah, interesting.
But this is something that I think, needs to be discussed, needs to be accounted for.
Yeah. I think this is really interesting. However, the first sentence of the abstract, there is a replication crisis spreading through the annals of scientific inquiry. I think that's a bit much. Okay.
Yes.
In some parts, there's a problem, but not everywhere. Right. Okay.
My prediction is that somewhere down the line in your NIH proposal, you're going to have to include something about the experimenter's gender.
I'm out of here.
Just saying.
Yeah, you're probably right. You are probably right. Rich, what do you...
The response to this may not actually answer the problem, but yeah.
Rich, what do you have?
On the same sort of gender bias scheme or whatever, I just ran across a very short clip that's on, there's an NPR Science YouTube channel. Okay. That's, we've had a couple of pics from it before, but it's an interesting channel all by itself.
Is that SciTech?
Anyway,
no, that's not SciTech.
At any rate, it's just a really short clip with an animation of a story by a woman named Ayanna Howard, who is a world-class roboticist from Georgia Tech, who's had a number of different projects in her life, among which were working for NASA to design robots for surveying other planets and stuff. And she got acquired a job one time to lead a project to design one of the first robots to do this kind of stuff, and describes showing up for the first day of this and walking in the room where they were supposed to meet. And there was, I don't know how many people were there, but there was a guy there at least who took a look at her and said, "Oh, the secretary's meeting has been moved. It's down the hall."
and then
she responds by introducing her. Go ahead.
I was going to say, yeah, I think you should just let people watch it, but because it's kind of like the book report versus telling the whole book.
Fine. And
we can tell it next week.
Okay. Good enough.
Reveal it.
That's good. I'll go for that. This is just one little bit. She calls this the courage to invent. And this was a moment that it, in her mind, took for her, courage to get through. And her message is that it does take courage to move forward and do innovative things. And so go for it. Find the courage.
It's good to tell your stories. I know someone who once interviewed for a position in science, and the chair said, "I'm not going to hire you," because she said, "Why?" Because you're just going to have a baby and go out and, doesn't do any work. Can you imagine? It's the way.
Yeah. And research shows that women who are mothers are at least as productive, if not more so, than women who are not mothers.
Really?
And and there's a
by
mother
definition,
penalty.
they're more productive.
And there's a mother penalty and a father bonus. So men who are fathers are deemed more stable, more likely to be productive, and so forth than bachelor males.
I think I'd be more stable as a bachelor than I am now. Kathy, what do you have?
I picked something not gender related. It's just a video about ice cliffs forming over the weekend. It's Vimeo. It has a soundtrack that I didn't think particularly added to it. The only downside is it doesn't tell where this is, but I looked at some of this guy's other videos, and they seem to be related to Lake Huron. So it's just a really cool waves and ice. It's not very long.
Very cool. Yeah.
Cool.
Well, we all have this gender arc without even looking at each other's picks today.
Yeah,
It's all coincidental. Very interesting. So I picked an article which goes back to our guys arc, and it's an article in Lifehacker by Lee Anderson. It's called Set Your Autocorrect to Change Guys to Folks. I don't know if you remember, but someone suggested folks. A few people have suggested we use folks instead of guys. And it's funny, ever since our discussion here, I'm very sensitive. And I hear women saying guys to other women often. It's just,
right.
I'm sensitized to it. And so this is a cool article about this whole issue. And there's a podcast associated with it, which talks about whether guys is universal. And this one person has just addressed it by using an autocorrect.
Folks and dolls just don't sound right to me, though.
But I'll let you in on a little secret. What you just commented on, Vincent, where women talking to other women might say guys. It's also okay in most cases for women to refer to each other just in the company of each other as girls. Or for me to say to somebody, "You go, girl." And if a male said that, probably wouldn't go over okay. But so that doesn't surprise me. There's a tweet quoted in the middle of this article.
Yes.
But the Twitter handle is Man Who Has It All, and he says, "I'm not hung up on the generic label chairwoman because I know it covers both women and men, comma, Toby, male chairwoman, age 56. So, yeah.
Pretty funny.
Again, welcome to our world.
And we have a couple of listener picks. Sal writes, "Hi, TWiV team, the famous podcasters. I was excited to see TWiV's name in the Washington Post. Congratulations. Who would underestimate the viruses? They can cause protests, global socioeconomic anxieties, and political unrests." Here's a link to how the avian flu led to what recently happened in Iran. And of course, if one runs a Google search, they can find abundant articles about the effects of climate change on conflicts and civil wars. Associations between climate change, agriculture, and re-emerging infectious diseases are becoming more and more difficult to deny or ignore. This is an article in the Financial Tribune. 100 million chickens infected with avian flu in Iran. And so far, 1.2 trillion reals, which is 27 million, have been paid to chicken farmers over their losses. Okay. As always, thank you for all the wonderful podcasts, not just TWiV. And Mark writes, "We want to get people interested in learning about evolution. If yes, then what better way than to use sex for motivation?" In the book Dr. Tatiana's Sex Advice to All Creation does this. It's framed as letters from different species seeking love advice written to the fictional Dr. Tatiana,
Great concept.
who answers in a way to explain how different species' sexual practices have evolved to create strange behavior. Here are a few questions from the book, questions in quotes. Dear Dr. Tatiana, I'm a queen bee, and I'm worried. All my lovers leave their genitals inside me and then drop dead. Is this normal? Perplexed in Clover Hill. Dear Tatiana, I'm a peacock, but I have a lousy tail. It isn't very big, and some of the eye spots are wonky. The whole effect is wall-eyed. When I put my tail up, the hens don't even bother to feign indifference. They don't look at me at all. Is there anything I can do to impress them? Invisible in Sri Lanka.
That's wonderful. I love it.
Great. Books available in print, electronic. Here's the link at Amazon. Thank you, Mark. That's very cool.
Can hardly wait to read the one from the praying mantis.
I think that's in there. This came out a while ago.
Very cool.
The female Black Widow.
I like it. All right, that's TWiV 477. I know they're long, but I think most people are okay with that. The people who object are in the minority. But you always have a fast forward button, right?
And the people who object aren't listening now anyway, so we can talk about them.
That's right. They don't get this far.
I found it amusing, though. We spent 15 minutes talking about how to shorten our show.
Yeah,
Yes.
that's right. Well, here's the thing. We could cut out the picks of the week.
Well, it's like the paperwork reduction notice on the bottom of a form.
Listen,
Exactly.
one of the reasons this is so long is because we're having a good time.
This is all
Well, that's
Okay.
it. That was my original
So-
idea, that we would just sit around and gab for a while.
That's right.
I was thinking about that early. Yeah, your original idea was as professors, we have these conversations in our offices,
Yeah.
and they ought to be published. I was thinking about that as I was looking up that mouse paper while we were on the internet while we were talking about these other papers. That's what we used to do all the time in my office. We'd come in and we'd start talking about something, and everybody would be Googling this and that and figuring it all out. It's great fun. Great fun.
All right. So that's it. That's what it's about. And I think some people out there listen. And I realize this will prevent us from getting really big, but that's okay. Right? We're big enough. If there are people listening, that's cool with us, at least for me. So we will just leave it the way it is. Microbe.tv/TWiV. TWiV at microbe.tv is where you can send questions and comments. Consider supporting us. Where else can you get 2 hours of unfettered conversation in deep science than here? Microbe.tv/contribute. Dickson Despommier is at parasiteswithoutborders.com. Thank you, Dickson.
You're welcome.
So your course just started.
Yep, just today.
Exciting.
Very.
Mine started on
Wednesday.
First.
Bright-eyed and bushy-tailed. I will never grow tired of seeing that. Honestly, I won't.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor at the University of Florida in Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
I bet you like having these chats because you don't have an office anymore for people to come in and talk
Oh yeah,
about stuff,
absolutely,
right?
absolutely.
Yeah. Alan Dove is at turbidplaque.com. You can also find him on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the introductory music, ronaldjenkins.com. Been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.